Hello again. My next two topics are complex, so I'll break those two closely related processes, photosynthesis and respiration, into two parts here. And we'll start with respiration. When we think of respiration, you and I probably think of the process we use to move oxygen into our body and release carbon dioxide. That's related to cellular respiration, but we're really going to be focused here on what's going on inside each and every one of our cells, inside their mitochondria. And this process of cellular respiration is one that's shared by both plants and animals. Both plants and animals, as well as all other eukaryotes, need to break down complex energy-rich molecules like the sugar glucose to harvest energy that can be used to do work. So that's what we mean when we're talking globally about cellular respiration. So I want to break this down with you into three critical steps. The first step in cellular respiration is called glycolysis. Glycolysis just involves taking that complex 6-carbon sugar, glucose, and literally breaking it in two. That's what the lysis tells you. We are lysing that molecule, splitting it in two. This takes place in the cytoplasm of your cells. It releases a little bit of energy. Cells can capture 2 molecules of ATP per molecule glucose that they break open. That's good. We need ATP to do all of the other work that keeps us alive and moving and warm, but 2 molecules won't be enough. So from there, those 2 halves of that glucose molecule are going on to step 2. Step 2 now takes place inside the mitochondria. So I've got a model of an enormous mitochondria here to remind us about the structure of these organelles. Remember that a mitochondria is a membrane-bound organelle that I might find in lots of different types of eukaryotic cells. While our textbooks and our models show 1 or 2 mitochondria per cell, there might be hundreds of mitochondria in each one of your cells. That's how important their function is. So remember, just to get our bearings, that mitochondria have 2 membranes around them made of phospholipids like every other cell membrane we meet. This first outer membrane is smooth. It defines the outer edge of the mitochondria. The inner mitochondrial membrane, shown here in white, is heavily folded. You'll sometimes see these folds called cristae, these inner folds on the inner mitochondrial membrane. This is important. It creates 2 things. First, it creates a lot of surface area. Look at all that extra surface area we have available for this chemistry. Second, it creates 2 discrete compartments inside the mitochondria. So you have this solution inside the inner mitochondrial membrane. It's called the matrix. And then you have a separate space here, a separate solution between the outer mitochondrial membrane and the intermitochondrial membrane. We call this the intermembrane space. So we'll be looking at some chemistry that takes place that utilizes both that surface area and those 2 separate compartments. So we're ready for step 2. We broke our glucose molecule in half, released a little bit of its energy in glycolysis. Those 2 halves of our glucose molecule will now be imported into the mitochondria. And it's here in the matrix that we're going to do step 2 of cellular respiration. This is known as the citric acid cycle, or sometimes called the Krebs cycle. In the citric acid cycle, a series of enzymes found in the matrix will completely break down that sugar molecule, releasing carbon dioxide. That's the carbon dioxide that you're exhaling right now, and capturing a little more cellular energy in the form of ATP. We again capture 2 molecules of ATP from this process per molecule glucose. That's nice, but that's not nearly enough to keep us going. So there's more energy in here available for us to do something with. It's just in a slightly different form. During this process of the citric acid cycle, not only have we been breaking down molecules, getting a little bit of ATP out of those efforts, we've also been capturing high energy electrons. Electrons can exist in several energy states, Some of them are in an excited form. These will normally fall back down to what's called their ground state. You know this, you've seen this if you've seen a molecule fluoresce, give off light as those electrons fall. You also know this when you wear a dark colored shirt on a warm day. The sunlight excites electrons, they fall down to their ground state and release that energy as heat, and you get warmer in your darker colored clothing. In the case of the cell, during cellular respiration, we're stripping off those high energy electrons from that sugar molecule, and we're using an energy shuttle, an electron shuttle called NADH. It's a complex molecule with a simple job. It captures high energy electrons in their high energy state and shuttles them to the final step in cellular respiration. This final step is the electron transport chain. So in your inner mitochondrial membrane of your mitochondria, you'll find a bunch of proteins in that membrane. These proteins are part of an electron transport chain. Essentially, we're going to take all that energy in that high energy electron and capture it. And rather than just letting that electron fall to its low energy state, we're going to do it in a series of controlled reactions. Anytime you're handing off electrons between one molecule and another, you're doing what's called a redox reaction. Redox stands for reduction and oxidation. Reduction is what happens to a molecule when it gains an electron. Remember that electrons are negatively charged, so gaining an electron is going to reduce your overall charge. If I gained an electron, somebody else, some other molecule, lost that electron. Losing an electron is called oxidation. So we always see those two going hand in hand. A reduction is accompanied by an oxidation. If somebody loses an electron, another molecule picks it up. So that's really what's going on in this inner mitochondrial membrane. We are passing electrons down this electron transport chain, allowing those electrons to release their energy in a controlled fashion. And what are we doing with this energy? Well, hopefully we're using it to pump protons. Remember, a proton is just a hydrogen ion. We're going to pump protons into this nice confined space between these two membranes, into the intermembrane space. Remember that protons are charged. They can't, even though they're tiny, they can't freely cross a phospholipid membrane. They can't get through. So if you pump protons into this space, they're stuck. And what you're doing while you're pumping then is building a high concentration of protons inside that space. I'd encourage you to think of those protons in that inner membrane space just like water behind a dam. There's a lot of potential energy there. You know that if we opened a turbine and a water spillway for the water behind the dam, if the dam cracked, all of that energy would come out spontaneously. We would convert that potential energy to kinetic energy. Same thing here. If you poke a hole in this membrane, those protons that you've been working so hard to pump into that space are going to spontaneously flow back out from an area of high concentration in the inner membrane space to an area of low concentration in the matrix. Now, instead of poking a hole, we're going to run those protons through a molecular turbine. This molecular turbine is an enzyme, a protein, called ATP synthase. The ACE on the end, ASE, tells you it's an enzyme. If someone named it kindly, what's in front of ACE tells you what it does. So in this case, this is an enzyme that synthesizes ATP. So as those protons come spilling out through our enzyme ATP synthase, it is going to take low energy ADP and convert it to our high energy useful molecule ATP.